Today, we are at the Institute for Cancer Research, where we will visit the Drosophila facility that Tor Erik Kristen set up here. I actually work here, so I know the place well. You know fruit flies because you spend your summers trying to get rid of them, where they feed on rotting fruit in your kitchen. While annoying at times, the fruit fly has actually contributed to our understanding of genetics today, which was elucidated by the work of Thomas Hunt Morgan in the 1900s. Work using Drosophila has led us to understand the genes that are important for development, and many of these genes have in fact turned out to be relevant for cancer. With its short life cycle, no ethical requirements, and low running costs, Drosophila is one of the most common model organisms used in the world, with more than 3,000 registered labs using Drosophila. Today, we're going to show you how to feed them, how to cross them, and we're going to talk to an expert who's going to tell us about the fly's applications. This way. Hi, Ricci.
Hey.
You ready to go and look at some flies?
Yes.
So now we're going to go in the fly room. You know this room really well,
yes,
since you flip the flies all the time.
Hours and hours.
So one of the great advantages of the fly is that they don't take much space, right?
No.
So we can have a lot of them stored in a small amount of space.
So they are stored in this kind of box, and there are always 50 different ones. When they need new food, you have to flip them, which is basically feeding the flies. And then you have new food, which is here. And we have like this potatoes, sugar, yeast, and all the good stuff the flies like. And then when the flies need new food, because it is old, then we just take a new vial with new food, and then we flip them over into the new vial. So the adult flies go into this vial.
So the tapping that you're doing, tell us what the role of that is.
The tapping is basically just because flies can fly, so you have to push them down in the vial so they don't escape. And then I transfer them over into the new vial and close them.
So have you ever,
and then you can just put them back.
Ever lost flies?
Oh, of course.
Yeah.
Everyone does. Normally they don't escape, but sometimes they do, but that's fine.
So one of the really nice aspects about the flies is that you don't have to genotype every generation. With the flies, we can really follow traits. You can follow your gene by because maybe it's attached to a phenotypic trait like the wings being curly or the color of the eyes, and so it's really great and it goes very fast. Right, so we're going to look at males and females, and a very important aspect about setting up crosses is that we need virgins. So I know it sounds weird, but we need the mother not to have met a male before, because we want to know who the father is in this case, because we're interested in the offspring. So let's have a look at these flies.
Okay.
So one of the things that we always need to do is to put them to sleep. As you know, flies fly, and so we actually just use CO2 to put them to sleep, and so it happens really fast.
Wow.
There we go. And you can see that they all fall at the bottom of the vial. And then I've also got CO2 coming into this little platform here, so they won't wake up and start flying around in the lab.
So now we have males and females?
Yeah, please. So we're going to have a look at them under the microscope, and it's very easy to see. And so I'm going to show you the differences. So virgins need to be picked early in the day, so we're actually a little bit late already. But what we can see here is that this is a male virgin and this is a female virgin. So they're much more transparent than later on. You don't have the typical black pattern that you have in adults. Yeah. And you can already see here, so these are only, you know, maybe 8 hours old or something like that. You can see the female is already much larger. And so the reason we can see the difference between genders is the size. You can also see this black area here, and so that's for the male. Here we've got adult flies, and you can see that the female is again larger, but there's no black posterior end, whereas here this is completely patterned. Half of this area is completely black, and just to confirm it's a female, it is also laying an egg. So that's males and females, and then the virgins.
What are we going to do now?
So I thought we'd go and see if Sabi is in the fly room.
Okay. And
so he's worked with Drosophila for a while, and he's going to tell us about the different applications.
Okay, so that's the fly room.
That's right.
Cool. Hi, Sabi.
Hi, guys.
What are you doing?
Oh, I'm just collecting some virgins. I want to do a new experiment and I need to set up new crosses.
Okay.
So you've been working with Drosophila for some time. Can you tell us a bit about your favorite aspects of the model organism?
Sure. Actually, I have been working with flies for more than 10 years. And I think the best thing with flies that you can combine the classical genetic approaches with modern molecular genetics or cell biology or even physiological techniques. And it's relatively easy to combine these things.
So what kind of diseases can one model in the fly?
Actually, flies have well-developed organs which are easy to compare to the human organs. And these organs behave quite similarly like the human ones. For example, inappropriate diet can cause heart failure or diabetes-like phenotypes in flies. But they can also develop tumors, or aged Drosophila, like old men, can suffer the loss of neuromuscular function. So there are a big variety of diseases.
Oh, that's so cool. But is there anything bad with the flies?
Oh, bad? No, there is nothing bad with the flies. I really love them. Of course there is. Regarding the diseases, so it's cool that the fly genome encodes homologs for almost 70% of human disease-associated genes. But of course, there is this 30% remaining. Another more specific thing, if you would like to study adaptive immunity, flies are not the best. They are invertebrates, so they don't have adaptive immunity. But they have native immunity, they have macrophages, they can undergo inflammation, but they don't have lymphocytes, for example.
So you mentioned tumors in the fly, and I know that you work with cancer. So how do you turn a normal Drosophila cell into a cancer cell?
First, I would like to mention that to study tumors, you need to do crosses, because if you have a tumor-carrying fly, then it will die soon. But if you cross a fly having an oncogenic transgene and with another which have an activator transgene for this oncogene, then in the offspring, you can have tumors which you can study. The superpower of the Drosophila that thanks to the powerful genetic system, we can generate mosaic tissues. It means that only a subset of cells will express these oncogenes and develop tumors.
So you're saying that you're really able to have cancer cells surrounded by normal cells and look at the interaction.
Of course, so the rest of the cells will serve as microenvironment, or if you don't study tumor setup, it can serve as controls.
And are you able to turn on your mutation of interest anywhere in the fly?
Sure. So this is also a great thing in Drosophila, that a lot of driver lines are available. So literally you can induce the expression or downregulate the expression of any gene in almost any tissues.
Pretty cool. Okay, thanks very much.
You're
See you
welcome.
later.
See you later, guys.
See you.
See you.
The fly is great, I think that's clear. With contributions to research for over 100 years, Drosophila has the advantage of advanced genetic tools. Today we have learned that the fly can be used to study many diseases, but here at our institute, the focus is of course cancer research.